And all right, so good afternoon, everyone. My name is Nati Wong. I'm a PhD student in Julian Schroeder Lab. So am myself is a plant biologist. Quite often in the interface, student talk, we often listen to like a whole organism, like ranging from a single molecule to the whole bodily function, right? Jack is really the pioneer in the field of microbiome and others research. Right? It's going to give us a different views of organismic interactions, and he's mostly study microbes and others. He also just mentioned that he study humans and also plant and others too. So yeah, without further ado, so let's welcome Dr. Jack Gilbert from SIO.
Thank you. I'll stand up if that's all right. I'm actually primarily a professor of pediatrics. I run human clinical trials, mostly focused around nutrition, food as medicine, that kind of stuff. We're currently running 2 clinical trials. One is focused on trying to determine if what you eat affects your physiological response. So I put down those chips, they're going to kill you. But the water's good and the salad's okay. And then the other one is how we can influence what you eat in order to prevent the risk of cancer development. So again, everything you're eating is probably good feeling, sorry. But it's going to be okay. But today, I have no attention span. I kind of dance around many different studies. So I'm also Deputy Director down at Scripps Institute of Oceanography. That's the Marine Science Campus on the beach. And I down there, I'm also Associate Vice Chancellor, supporting research operations, managing faculty. We have over 200 faculty at SIO. Come down and see us. It's a school of the university, but it also formed the university in the 1960s. So it's been there since 1902. A very old, august organization. So from this perspective, I get distracted really super easily, and I go off on crazy tangents. So if I do, I apologize. But one of these tangents was in soils. And I used to work at University of Chicago for about 10 years, where I was actually a professor of surgery. They didn't let me cut anyone open, but I did a lot of surgical praxis research. And when I was there, I also got interested in how to make food more available to the community. A lot of our communities were food insecure. And so I got involved in building local farms in and around South Chicago and empowering local communities to lead those farms so that we could actually have healthy food available to people and that they would have ownership in the generation, selling, and distribution of that food so they can make a profit of it as well. So I got super involved in that and thought that'd be a super cool way of bringing microbiology and microbial engineering into that practice. I'm definitely a translational scientist. I like seeing how you can make something out of what we create. So basic science is kick-ass, translating it is kick-ass, and making it work for the world around us is also key. What else do I do in that context? So I'm going to talk to you today about Unji Soil Health Centre, which just started up last year, and its implications and interactions with human health, because there's very unusual ways in which soil health can actually influence human health. And but also I want to highlight the fact that a lot of this is predicated on our ability to get technology out there and make it available to people. And that means making sure you understand what an invention disclosure is, making sure you understand intellectual property and IP law, getting involved in reaching out to the broader community, not just the original stakeholders that might value your scientific input, but also ensuring that you as individuals have the ability to translate and push science out there through startup companies, through partnership with private organizations, break outside of the ivory tower. And I want to make this tower very porous, so it has the opportunity to actually serve the community in which we live, right? Not just the wealthy. Okay. You also notice I'm British. I'm now British American. I got my citizenship just in case. And but as an immigrant, I've also been in this country now for about 17 years, and I do kind of love it. But San Diego is a lot better than Chicago. Just saying. Okay, cool. So this is a presidential action that came out about a month and a half ago. we're dealing with a very, very complicated socio-political environment right now. And if you anyone harks back to about the 1950s and Kuhn, his idea of paradigm shifts. If you don't know what that is, look it up on Wikipedia. Paradigm shifts are embedded in the principle that we as scientists, as educators, as researchers, don't exist in some kind of weird vacuum. We exist interdigitated with the community, right? We are the community. And so any research that we do is embedded in that reality, right? And we are never unbiased, no matter how much we tend to be biased. Human beings have biases. I mean, it's shock horror, right? That's the entire benefit and also detriment to the entire species. But those biases place us in the context of whatever socio-political environment we exist in. And our current one has complexities, but it also has some weird good bits in it as well. So this is a presidential decree. No, not decree, what are they called? Not allowed to call them decrees because that would mean that we were living in an autocracy. They are presidential thingamajiggers. Yes. And this is executive order, thank you, is a mechanism by which the president can put in place pressure on the legislator to change how they deliver resources and funding. So this one came out, advancing regenerative agriculture, strengthening American farm resilience, regenerative and resilience being the two key words there. That's weird as, because we've been trying to do that for like 20 years and not being able to get much traction on it. Now we have the president of the United States saying this is something we really do need to do. We need to make American agriculture more resilient. Food security is national security. I'm using all these words deliberately because that's how you communicate when you're up on the hill, up in Sacramento, when you're actually interacting with people either side of the aisle. They want to hear you saying certain pitch words. Those are the pitch words which we're using. So I will integrate them and splash them throughout this presentation because you are not living in isolation. You're living in a world of constant integration with the world. Okay, plants do the same thing. So plants exist and have evolved in a sea of microorganisms. Over the last about 175 to 285 million years, they have existed and created their own structures to survive in a world that's microbial. 4 billion years of microbial evolution and maybe half a billion years of macrobial evolution has led everything around us, including you, being entirely dependent upon that adaptation mechanism to leverage and work with the microorganisms which you find yourself interacting with. And plants do that by pumping a bunch of crap below ground. So they use their little photosynthetic leaves, they make sugars, they make carbon products, and they pump them through their roots into the soil. And they use that to attract microorganisms they can use. You do the same thing. You eat food to actually feed the bacteria in your gut, which then produce compounds which your body needs. Short-chain fatty acids, indoles, which have become incredibly valuable. Our bodies become dependent upon those compounds for supporting our overall health. We actually do a really cool project in a moment in Jamaica where a lot of people, our protein is sufficient in the rural community. Protein sufficient. Their bacterial community is actually providing a lot of the protein that helps their bodies grow. And so that's why some people are resilient to protein depleted diets and some people are not, because you've got the right kind of bacteria inside your body. Again, I do tangents, I apologize. But plants are harvesting, growing, agriculturally sustaining their own microbial garden in order to support themselves, to enable that they can get access to different nutrients, which they wouldn't be able to get access to without the bacteria. They can help, the bacterial relationship can help with stress resilience, competing against pathogens in their environments. It can help with access to water because bacteria and fungi pump out all these gloopy compounds into their environment, which help retain water into the soil around the root. They can even be a source for the bacteria that get inside the plant and get up into the fruits and the leaves. And they can help to structure the soil in a way that enables the plant to rapidly grow. We're actually doing a really cool study right now with the Californian legislator. We have several very big programs that we run where we are taking burn scars, so areas that have been recently devastated by fire, which actually sterilizes the top 5 centimeters of the soil. and we're literally using planes. We're spraying that area with bacterial communities and small seeds of native plants. And what we found is that the seeds and the bacterial community rapidly accelerate the restructuring of that soil, preventing that soil from slipping off the land and causing widespread devastation to local communities. Also improving water retention, etc., etc. So these can be incredibly powerful things. Oh,
You just want this out of the way?
Sorry, I was just gonna ask him to move that,
oh, sure.
so
Yeah,
we
you
don't,
go. Do you want to do it flat? Then you just get that one.
there, we were missing.
I can even just get rid of the entire floating commands if you want.
Yeah.
That way, we don't miss any of his
Okay.
slides. Thank you, Doug. Sorry.
That's right. Just ping me. I'm also. This is what I do for you. So soil functions are incredibly important for sustaining life on this planet. From water purification, we actually think of the soil as the world's biggest water purifier, water filter, to carbon sequestration, locking carbon from the atmosphere below ground. The ocean does more of it, but soils are incredibly important. Provision of food, fiber, and fuel for humans. Cultural heritage is a key one that most people ignore. Soil is part of our history, it's part of our culture, and many communities around the world, soil is incredibly important. Ayurvedic communities in South Asia revered the soil as a part of their ancestry, were incredibly supportive of soil development in their ancestry. Chinese people, in around 1000 BC, used to take soil in little containers from their hometown where they grew up before they were shipped out to different parts of the empire to do administration, because they believed that they could cure illnesses if they got them far away. So what they do is they felt sick or maybe homesick or actually had gastrointestinal illness. They take some of that soil from their home community and make it into a tea, which they would drink. And we think now, and I'll point that out later, that might actually stimulate your immune system with microorganisms and antigenic adjuvants that you've seen growing up in your immune system when you were young that actually support your health later on. Interesting. Got a lot to learn from people. Soils are under multiple threats. Sorry, there's lots of other things you can read there. And multiple threats that's actually massively impacting food security, which is national security, remember. Climate resilience, you can drop the climate and just say resilience. National resilience at a scale. And agricultural practices are significant contributors to water pollution, greenhouse gas emissions, and they're causing many additional stresses on our environment. Especially in San Diego County, we have the largest number of smallholder farms, actual smallholder farms that are financially productive anywhere in the entire US. And that's mostly because of our geography, and it's kind of hilly and weird, and we don't have much water, so people have to build small farms. You can't have massive row crop farms. But that's meant that our environment is actually very, very amenable to change in agricultural practice, but also has a lot of difficulties because those local farms can produce a hell of a lot of local pollution, which can damage river systems and water systems in our areas. Soils are under massive threat. Estimated 33% of the planet's soils are already degraded to some degree. And most of this is due to massive over-harvesting of that land and pushing that land to produce more for less, mostly through use of synthetic fertilizers. We just keep on pumping in nitrogen, phosphate, potassium at levels which are unsustainable. And that's just leaching nutrients out of the soil to the point where the soil can no longer produce the same kind of crop yields that we were getting in the past. We think we've got about 60 harvests left of our topsoil in most of our major agricultural regions. That's about 60 years worth of productivity in many of our major soils. And these are our big bread baskets are in, for example, like the Ukraine, northern China in an area called the Black Soils region, and then in the central US in the prairie regions. These are areas where you used to have 15, 20, 25 feet of topsoil, and they've been slowly compressed as we've eroded them or destroyed them. In those areas, we are running out of soil. Where do you start with this problem? You start with the farmers. This guy's name is Adam. He actually works up in Encinitas on a farm called Coastal Roots Farm. When we wanted to explore how to actually approach these kinds of problems with our science, And we could have done that just by doing the science and then going and creating a little company and then spreading it out there. Or we could go and talk to the farmers themselves and say, hey, what is the problem that you're dealing with? And that's what we decided to go with. We said, hey, we got cool technologies, but we don't know if you want them. We don't know if they're deployable in your environment. And we don't know if they'll actually solve the problems which you're interested in. So we went and created a community research integration program with social scientists at UCSD to support our integration into that community and make sure that we were listening to them. And their big concerns were input costs. Synthetic fertilizers going up through the roof, especially now with even with Hormuz being quote unquote open. Well, there's a lot of insider trading which might need to be done in order to make more money off of that before we see it actually fully open. Didn't say that out loud. Your responses to NPK are going down. He keeps on saying, our farmers keep on telling us that, hey man, if we keep on putting in this fertilizer, we used to get these kinds of yields when we did it. We're not getting them anymore. Something isn't working the way it used to. And then regulatory pressure. Chemistries for both herbicides, pesticides, and fertilizers that worked 20 years ago are no longer working. But these mechanisms are leaving the economic market. There aren't ways to replace them easily, but regulations stipulate they have to be used. So the legislator in both California and local and federal state regulations, like in the Farm Bill, are not keeping up with the problems that farmers are experiencing. So we also figured out by working with our collaborators, the GPS, the paper I published with Daniela Faganini-Diaz and David Victor, that we can theoretically leverage existing agricultural systems to solve a lot of problems. So, for example, in this study, we looked at agricultural systems, big ones in, say, Brazil, Argentina, North America, Europe, South Asia, especially India and China, and said if we were able to activate the soil processes in those environments with plants, deep-rooted plants especially, and microbial biostimulants, adding bacteria back into the soil, what would it do for carbon drawdown? And we were able to demonstrate through a modeling process that we could get up to about 1.2 gigatons of carbon dioxide drawdown just using the agricultural land in those areas per year, which is 7 times the current offset market for all the CO2 drawdown in the entire world. Currently. So soils alone in just those countries and one continent would facilitate us being able to massively upscale the amount of carbon being removed from the environment. This is a case study. There's a case study of how to use existing economic systems and existing mechanistic systems to facilitate your outcomes. So, David and Daniela are economic modelers. Trying to understand how systems work. They don't know anything about bacteria, don't really know anything about plants, don't really care, but they do have a mechanism. That's the beauty of a university, is you can bring together weird people who want to actually talk to each other despite not knowing anything about each other's work. Okay, when we work with the biology in soil, the benefits are immense. We think of microbes as little farmers, and they're incredibly important. This is actually on our soil health center wall. In the lab down at Scripps Institution of Oceanography, microbes are the littlest farm hands. They're incredibly important. And they are important because they are the ones processing all the biochemistry in the soil. They support the plant recruits them because they're important to plant's activity based on evolutionary history, but they're also doing a hell of a lot of other things in the soil. Soil is there because of microbes. Soil is microbes. Microbial life is essential to soil development and maintenance. So we've been exploring different strategies, both probiotics, microbial inoculants that we add back into the soil system in order to promote it, either directly or through different strategies. We can take like an entire healthy soil and transplant it into a sick soil. I've done this with humans. We do a fecal microbiome transplant. Interestingly, this is another ancient Chinese remedy. It used to be called yellow soup. I don't know the exact Chinese pronunciation, but they would take the feces from a newborn infant and put a piece of cheesecloth over it, leave it outside overnight to ferment a little bit, and then feed it to people who had chronic diarrhea, and it would help to repopulate the bacterial population in their gut in order to overcome infectious disease. We do this now called fecal microbiome transplants to treat Clostridium or even difficile infections in people who are suffering from an inability to respond to antibiotic therapy. In fact, the only two clinically approved microbiome therapies by the FDA currently are for the treatment of C. diff, recurrent C. diff disease through those kind of mechanisms. But you can do the same with soil, take healthy soil, put it into diseased soil, and allow the microbes in the healthy soil to populate that soil environment, or we can add them in directly with literally a probiotic like you buy in the market, but we add them in, put them into the soil and plant system. Or we can use prebiotics, things which stimulate microbial activity. Exactly the same as taking a fiber supplement or eating a healthy salad, so I'm eating it. And it will help to grow the right kind of bacteria inside your body because that's bacteria food. It actually does very little for your health, but it goes straight down into your colon and it helps the bacteria to ferment and produce the products which your body actually needs. So this could be incredibly important. So we formed the Soil Health Centre around, this is last year, around the adage that we could work with the community to identify microbial solutions to support their needs. It's based around the idea that we can work across UCSD as an organisation to facilitate that kind of outreach, a porous community, a porous institution that allows the community to come in and work with us through biology, physics and engineering, chemistry, both soil and new chemical adjectives, economics and public policy, like I talked about with David Victor and Danny, and then social science and ethics, very, very key. You're going to work with the community. You've got to understand how to work with the community, and too many scientists are crap at doing that. We're just like, oh, I got a solution. Why are you using my solution? No, I'm not using your solution because your solution is inherently biased, or it has, significant problems associated with it, or you're not my race, and therefore why should I care? These kind of problems are manifest in our population because why? We are a biased species, socially and economically biased species. Anyway. So we are leveraging interdisciplinary expertise across these groups to develop new technologies that help us advance both climate smart agriculture and healthy soils. We don't say that to the federal government. And enhance carbon sequestration and reducing the need for agricultural inputs. Remember, that's one of the key things that the farmers said to help us to save money. We don't want to put so much stuff in the soil. It costs a lot of money. So if we can stop that, then great. We'll make more money. Building a translation engine to scale cutting edge technologies and actually bring them into the farming economic market. That means not just creating the science, but figuring out what science products that we create, how they can be translated into a market. Is there a market for them? Will people buy them? If we put this IP out there, will Big Ag just take it, stick it on a shelf because it actually disrupts their existing financial markets, right? These are all things you've got to start thinking about. Facilitating the adoption of sustainable practices by incorporating grower perspectives and feedback directly into the development of these products. That means bringing those farmers into the lab, showing them what we're doing, reporting back to them on a regular basis, getting their input, listening to them and actually engaging with their input to facilitate real outcomes. And so we've now got professors and researchers and practitioners from all over the university. Over 11 schools are represented. I think that's every school on campus now. And about 17 to 18 different departments across those schools that facilitate us being able to build and bring diverse voices into our structure, because diversity is a strength, no matter what the federal government says. And we are cultivating those partnerships with the wider community. We now have over 30 different local farms involved in our practice. They provide us with the opportunity to work with them to develop new technologies, which they own the IP for, so that they can make a financial profit off those margins. And we have very complicated relationships with lawyers to enable us to do that through UCSD. We have a lot of local organisations which we partner with to help facilitate this. A lot of these aren't new ideas, they're just new nuances on ideas. So working with, say, Salk Institute and their Harnessing Plants initiative, they build better plants, we build better microbes, good partnership. South Coast Research and Extension Centre helping us to reach out to the wider community. Local Indian communities such as Squam Band, who have been instrumental in helping us to bridge out across the rest of the Kumeyaay population and the Luswenyo nations to enable us to work with landholders in that area in a way that doesn't involve some white English guy coming in and saying, oh, I've got a solution. So we're engaging them in actually developing these technological solutions. White Buffalo Land Trials, natural communities, even the zoo. We're heavily involved in helping to support those initiatives that actually bring the value back to that community. And then wider consortia across the world. So I formed something called the Microbiome Sensors Consortium to allow us to be able to take advantage of microbial technologies developed at universities all the way around the world and bring them to bear where needed in our technologies. And then I've just formed a new species survival commission, but just for microbes. So you've heard of panda bears. There are 9 species of otter on the planet, right? Only 9. I have about 700 billion species of bacteria alone, right? It's a bit of a bigger problem. The otters have 1 species survival commission for 9 species. I've got 1 species survival commission for the entire microbial universe. I think my problem's bigger. But so we built that into the IUCN framework, the International Union for the Conservation of Nature, to bring microbiology into the conservation discussion so that we can conserve that microbial technological resource so that we can deploy it more effectively in these kind of economic systems. Okay, we're also working with developing new models and new mechanisms. So one of those is something we call meta-riboseq. So a lot of you might be familiar with one of my collaborators, Carsten Zengler, who's a professor in engineering and pediatrics. Got a kind of a weird double end thing. He's retiring next year, if anyone's not heard that, at the age of 59. Not sure what I would do. I had thought about it, but I'd probably just buy more motorcycles, which is not a good habit. Don't get into that habit. So we have 3 techniques here. One's called metagenomics, where we sequence all the DNA from a community and we try and recreate the genomes of each of the organisms. When you got soil, where we can have 1,000 to 2,000 species in a gram, that can become pretty complicated. But we can start to tease apart who's there and what their potential to do stuff is by looking at their genome. Then we can do and look at the transcriptome. That's the messenger RNA being transcribed out of the genome before it makes its way onto making a protein. This is valuable, but we found out that bacteria produce a lot of messenger RNA that kind of just sits in the cell in a pool waiting to be used. So when you sequence the transcriptome of a microbial cell, you're really looking at things it's thought to transcribe to create a pool of resources to allow it to respond rapidly when it needs to, right? So you've got a ton of mRNA that's sitting around, not being made into protein, not being made into enzymes, not being utilized at the time you assay it. So working with Carsten and a few other colleagues here, we developed something called meta-riboseq, where you look at the messenger RNA that's actually bound to the ribosome in the process of being translated into protein, right? So this is a different strategy. This is the mRNA, the transcriptional activity, that's actually in the process of being made into something. And that makes it much more informative about what's happening right now. And when we compare what's being transcribed out of the genome into what's actually being made into protein, we're able to get a much clearer picture of the translational efficiency of the cell, what it's prioritizing, what it's putting its energy into at that time, which helps us to really understand the communities of organisms which are important when we start applying bacteria into these environments. And so Vanessa Minnis here is very, very happy because her favorite bug, Mycobacterium, turned out to be incredibly important as a particular organism in the riboseq data. It was actively involved in doing stuff, even though it was very rare in the genome. It wasn't a particularly abundant organism, and the transcriptome said it wasn't really doing very much at all. But it's actively transcribing most of its mRNA and being an engine for metabolic change in that environment. So these are really cool things you can do. We're also leveraging AI and supercomputing. So we use a metabolic modeling system where we take the genome, the transcriptome, that riboseq, metabolomics data, where we look at the metabolite products using mass spec, and then we build that into a metabolic model. We say this organism has the ability to do X, Y, and Z. And then we put it under certain conditions, feed it certain compounds, increase the temperature, reduce the pH, change its environment, and then see in the computer what it does. And then we add a friend in, and we get 2 models interacting with each other. Does this, when I increase the pH, produce something which feeds this bug? And does that change their relative abundance based upon how much energy they put into cellular replication, right? Does that increase it? We call this mind. It's an ability to model the metabolic system using all this data so that we can predict how these organisms will respond to the environment in which they find themselves. And we've applied this now to the roots of plants because we've got a metabolic model for, say, a tomato plant looking at how it responds to the environment, what it puts out there into the world to recruit bacteria. And then we've got metabolic models for each of the bacteria to see how they will respond to that plant relationship. Using this, we can figure out exactly which bugs to add to the plant to stimulate a certain process in the plant, like increased productivity and yield, or changing the nutrient density of the tomato. And we can also figure out things to add in to stimulate the microbial community in a soil that might help. And so doing this, we've applied this to multiple farms around San Diego now. We went into 3 different farms and we looked at their different environmental characteristics like pH, which can vary from 6.4 up to about 7.6. That's a lot of orders of magnitude change. Or organic carbon percentage, or changes in organic carbon nitrogen ratios. And then we can use this information to design microbial communities, which we then add into this farming system and see if it improves the productivity of the crops. So we did this first of all by looking at whether just adding dead bacteria makes a difference to adding live bacteria. Do the bugs need to be alive to make a difference? And indeed they do. When you autoclave your microbial community here in blue, you see that we get significantly less growth potential than we do when we add living bacteria in. And that's important, right? Because bacteria are dead organisms, they've got a lot of nutrients in them. Maybe the plants just respond to them. They don't. The bugs need to be alive. That actually, sorry, tangent. Human probiotics, not enough people do dead probiotic trials. And I do a lot of stuff in gut-brain axis, mostly for Parkinson's, depression, and autism. And we found out that a lot of the probiotics that people feed to treat disorders or support the health journey for those particular conditions work just as effectively when they're dead as when they're alive. So it means that basically most probiotics are just stimulating the immune system as they pass through. The bugs don't need to get in there. They're not biologically active in the body. The immune system is just seeing them, and that's changing how your body reacts. Fun aside, I've got like 5 other talks on that. Anyway, and then we go in and we look at the metagenomes, metatranscriptase, metaribose, and we build a picture to try and understand how these organisms are surviving and what mechanistic role they're doing. And so, for example, we also explore how to what it needs to be inoculated first. So we have bacteria and fungi, for example, and in this situation, when we add our bacteria and fungi at the same time, we get significant improvements in the growth of lettuce. If we add bacteria then fungus, or fungus then bacteria, or just bacteria only and just fungus only, so that they're not being inoculated at the same time, like there's days between their inoculations, we don't see any significant increase in growth. So the partnership is important. That biological relationship in the addition is key. I don't know if you're asking a question or just scratching your ear. Cool. Initial results are pretty effective. We can significantly increase the root length of these crops in ways that makes them more resilient. They go and try and tap into water and nutrient resources deeper in the soil. So our studies have demonstrated that, for example, in lettuce, and I know lettuce isn't popular right now, every time people forget about the importance of microbiology, we release something like it's called lab release. You might have heard about it in the media. We release something in the community to make you realize that microbes are important. Then you hire more microbiologists to solve the problem. It's a big microbiologist conspiracy, but we just haven't told you about it yet. But we add these bacteria in. They stimulate the crop to produce big, deeper roots. That makes the crop more resilient. And what we found is that this means that the crop it requires less water input. So we can reduce the irrigation for this farm by 30% and get no yield deficit. So the plants still produce the same amount of lettuce. And we've done this. I also am a chief advisor, scientific advisor for an organization called Oath, which is a public benefit corporation. Weirdly, our primary donors are the band U2, and so Edge and Bono and team, and then a whole bunch of actors from Hollywood who are good friends now and are very engaged in trying to improve soil health. So we've taken their money and lack of sense and translated it into science and are now deploying it at scale. And one of our key relationships is with Rwanda, which is a tiny little country in this middle of Africa, run by a semi-demagogue, but a person with their heart potentially in the right place in this context. who is very interested in taking these microbial technologies to wean the country off of the need to buy synthetic fertilizers. This is important. There's colonization, historical narratives which have made this necessary, right? You know, Europeans have made farming communities in Africa addicted to utilizing these products, and then they take them away as soon as the price goes up and they need them more, right? So then the people that get less fertilizer first in a fertilizer crunch are African nations. And so President Kagame's had enough of this. And he basically came to us and said, hey, you know, can you come up with a different solution? We don't necessarily want to just build more fertilizer inputs. We want to create different solutions. So he's put $100 million into building a microbial technology platform for the entire country. And by 2030, we'll be in 93% of all agricultural land in Rwanda, which is huge, right? We've shown that we can replace fertilizer inputs with microbial biostimulants and have a yield improvement or yield benefit while regrowing the soil and making it more resilient to change, especially climate change, which he knows is coming. Sometimes demagogues can be important if their heart's in the right place. So here, for example, working with maize, we see significant improvements when we use our biostimulant techniques in yield, in every single area that we went into in Rwanda. And this is key because different areas have different climate practices, different soil health strategies, baselines, soil qualities that are incredibly important. And you can see there's a mountain range here, right? So there's a precipitation gradient across the country. Okay. We've done with tea as well. As a British person, I had to look at tea. Tea is important to our entire culture. That's the only reason the British Empire ever existed, was to get tea from the rest of the world. That was a bad joke. But in this context, we saw, could you reduce the amount of nitrogenous fertilizers, NPKs, that we add into tea production? And could you replace that with a biostimulant? So this is a really, really weird graph. Doesn't make a lot of sense. But here on the right, I'm adding a standard amount of fertilizer, so 350 kilograms per hectare of synthetic nitrogen, phosphate, potassium fertilizer, right? And that's the yield in terms of fresh weight of leaves produced by the crop. Here I have 70% of the nitrogen input. Here I have 100% of nitrogen input. Here I have 80% of the nitrogen input, and here I have 50% of the nitrogen input. And you notice it's going up. None of these are significantly different from each other, so I think that's a statistical artifact and I need more data. But each one of these has our microbial biostimulant produced and added in. So you can reduce the amount of nitrogen input by 50% and still get more yield when you add our biostimulants in than you do just with using the amount of fertilizer, which is important, right? So you can wean an entire country off of a harmful substance. So farmer wins first. We reduce the input costs for that farming community, the reliance upon international markets, which are biased, racist, and developed by European architectures. We can increase the risk of generating yield in difficult years. The entire of Europe is currently in a massive drought. They've been in a drought for nearly 8 weeks, which is unprecedented. And so they are panicking. But most of our African nation communities have been under varying droughts for a lot longer than that. I know we'll give a crap. Premium market access is a potential. A lot of our farmers, especially in San Diego, believe that by switching over to biostimulants versus using traditional methods, they can get access to a better market, sell their product for more. Not necessarily what we want to improve food security for vulnerable populations, but if you're working in La Jolla, who gives a crap because they've got enough money, they can pay for it. And then a reduction in regulatory exposure, because this is just microbes from the soil that the plants are grown in that we concentrate and put back into the soil that the plants are grown in. So there's no regulatory burden, and the EPA and the FDA agree that we're just literally using soil fertilizing to do it. It's great. So if this works, why isn't everybody doing this? People tried for 80 years. The idea of taking Bradyrhizobium, for example, which is a nitrogen-fixing bacteria, and adding it into a legume to produce more productivity by increasing nitrogen yield, is been around for a long time. But many people jumped on this bandwagon. People wanted to do GMOs. People wanted to modify microbial communities, but just use one or two organisms, whatever they could get off the shelf. And none of it really worked. The farmers got a whole load of stuff that they call bugs in a jug. And every time you go, oh, you're not a bugs in the jug salesperson, are you? I'm like, no, yeah, but no. So that black box and the cowboy Western nature of the industry meant a lot of our agricultural people are just tired, and they feel it's they've been burnt out by people lying to them. So we are literally going from the ground up and trying to rebuild faith in these products. So I did say healthy soils produce healthy people. Have I got enough time? I don't care. I'm going to keep on talking. I do, so it's 12:42. Kids that grow up living and interacting with healthy soil are healthier. And how do I know this? So again, I was in the Midwest for a decade, and my research out there ran something called the Dutchess-Well Family Institute, but it was focused around microbial technologies to support people's health, especially kids. And we worked with communities all across the Midwest. This is an Amish community in Indiana. And there's a weird health-based dichotomy between the Amish and the Hutterite communities in South Dakota. Both of these populations come from Central Europe around 150 years ago. Both of them eschew technology in the home, right? So you don't have any cell phones, no iPads, no computers. But they differ in one major respect, or two major respects. First, the Hutterites have almost a quarter of their population developing allergic disease, atopy, mostly asthma, a high risk. The average for the US is about 8%. The Amish have only about 4% asthma risk in their population, so significantly below the US average, half of the US average, and almost 5 times less than the Amish, than the Hutterites. So what's the major difference? The Amish live on their farms. The kids grow up living on their farm, working on their farm. They are 5 feet from the barn door to their front door. They don't have any technology, so they have to do work on the farm. Anyway, they're interacting with the farm on a regular basis, right? The Hutterite kids are not. You have to be a boy over the age of 14 to be allowed onto the farm. And so no early life exposure in either sex is allowed, which creates this problem, right? We think that was the hypothesis. So we did a lot of testing. We do a lot of work with animal models. So sorry if that is a concern, but mice weren't necessarily harmed in the process of doing this. We actually made better. So we took a mouse and we made it have a sensitivity to a food allergen called valvemin, to egg protein. When the mouse gets exposure to a valvemin, they get a massive eosinophilic burst in their lungs, akin to having an allergic sensitivity response, right? Not necessarily enough for an EpiPen, but enough to make you feel like you're having hives or reactivity. And we can see that when we take bowel lavage, so put liquid into the lung, pull it out, like we do with children, and we can look at the immune cells present in that material, and it shows us, yes, the lungs are under inflammatory response. If I take those same mice and I expose them to dust collected from the bedrooms of the children, I literally know, I mean, it's like reverse Grinch. We went in and we used vacuums in their bedrooms. This is the place where they're breathing most of the dust into their lungs deep, where they're sleeping, right? Key. We take that dust and we expose it to the mice through intranasal instillation through into their respiratory system. We see a significant reduction in the eosinophilic response when we expose them to a valvemin. Not so for the Hutterites. Hutterite kids' bedrooms are nowhere near the farming environment. They live in like this little commune away from the farming community, and the kids aren't allowed on the farm, so the kids never get it on their clothes, in their hair, in their respiratory tracts. They don't bring it home. The dust from the kids' bedrooms in the Hutterite community was not protective. Many of my Amish friends said, "Oh, dude, can we start selling this dust?" And I'm like, I don't know if you want to, but I don't know how you get that past regulatory approval. But the key thing is the kids, we're just literally seeing an allergic immune-based response to that capability. And so colleagues of mine took this. I was thinking of moving to Finland at one point. So I helped set up a research program over there, and then I didn't go to Finland. I went to San Diego, which was better. But maybe, I don't know. There's Carsten, who's been involved in this program, for anyone who cares. So what we did in Finland is we said, okay, if healthy soil is so good, maybe we can randomize an RCT, or a randomized controlled trial, to determine if that exposure makes a difference. And we took soil from the communities, the farming communities, the urban environments, ground zones that have damaged urban soil, and then from the forests. And we randomized the children to preschool, so 3 to 4 year old kids in preschool. We randomized them to interact with one of these pots for about an hour a week through dirt play, right? You know, mud pies, etc. The kids that interacted with forest soil had a significant reduction in inflammatory baseline. Their systems were less inflamed, their immune systems were less reactive. A significant reduction in metabolic disease risk by the age of 5, and weirdly, a significant improvement in their school readiness scores by the age of 5. Right, so just interacting with quote unquote healthy soil seemed to play a significant role. And so we in the States woke up to this idea about 2 years ago, and we've launched a double-blind placebo-controlled clinical study. It's not really placebo-controlled. Well, it is, but not in a good way. Where we take children and we randomize them to receive either an outdoor education or a urban preschool education. In our preliminary studies, we get the same kind of result. The kids getting the outdoor preschool education have significant reductions in allergic disease and significant reductions in metabolic disease by the age of 5 and significant improvements in school readiness scores. So playing around with good dirt makes you healthier and happier and potentially more intelligent. I don't know, I may be overselling that, but you get the idea. And as I said before, we built a species survival commission now for microorganisms. If these microbes are so important, maybe we should be considering them when we consider conservation practices. I do a lot of work with animals as well. I did a dolphin fecal microbiome transplant back in the day, which was fun. Dolphins get diarrhea too. But the bugs that are present in the dolphin are not kept anywhere, right? We don't keep a collection of dolphin-associated bacteria. We just finished another one where we got the baby penguins from Birch Aquarium, little baby fluffy baby penguins, and we showed that they need mother's milk to actually survive effectively with their interaction between their microbiome and their immune system response. And we're trying to create a library of all of the bacteria that are present in these penguins. Why? Because the bacteria are pretty much ubiquitously distributed around the world. I mean, everything you can see your eyes, all the macrobes are kind of boring. They've been around for a little while, but not very long, right? And they're very, very low diversity. If I take E. coli, there are 67,000 genomes of E. coli available in the public database. They can differ in their genetic coding regions by up to about 60%. If I took any one of you and I swapped out 60% of your coding regions in your genome, I can make you into a fungus, right? So one species of bacteria, we have the same genetic diversity as pretty much the entire kingdom of eukaryotes, which is insane, right? That's a massive over representation, but it's a fun stat. Okay, so what do you do with that? You want to collect and preserve as much of that microbial diversity as possible because nature's better randomizing and sorting and figuring out what works than any of our AI platforms and any of our AL systems are. So we use that system effectively to collect as many as we can. This works for humans. There's a bug called Bifidobacterium longum infantis, which is present in babies' intestines. Mother's gut actually accelerates the feeding of this bug in the intestine in the 3rd trimester of pregnancy, so it becomes more abundant. And then we hypothesize, we have some data from animals, but hypothesize that it's then selected from the gut via translocation into the lymph system and then transplanted into the breast ducts to produce a probiotic, prebiotic formulation for breast milk. Because breast milk has its own bacterial population, and we have a team here on campus that does a lot of this work in breast milk research. The bacteria that are present differ in different cultural communities, right? So Bifidobacterium longum infantis strain X is different for white European communities compared to women rearing children in Ghana, right? So you have to figure out, does that make a difference? If we created a monoculture of Bifidobacterium longum infantis and fed it to the world, would that solve the problem? Or is that genetic diversity inherently important in the health and regulation of all these peoples that have grown up around the world? I hope the latter, I think the latter. So we're trying to create a conservation library of all these strains around the world. And we're putting them in a giant vault that I can't tell you about somewhere in the world. called the microbiota vault. It's like the Svalbard seed bank, but it's got bacterial populations. And the idea being that if the great orange one does something really, really dumb and causes a world war, there'll still be a collection of these organisms that we can use to rebuild humanity. And if you are interested in microbiology, I'm also president of something called Applied Microbiology International. It's the world's second oldest microbiology society. So societies are really important to get involved with, especially early in your career. I did, and it's been massive support to both my mentoring role, my career development, and also you can get free travel grants and free funding. So if you're interested in microbiology, join AMI, super cheap to join as a student for a year. And you get access to a lot of cool perks. And also you can get cheaper publishing through our journals. And if you are interested in microbiology, go to this website. This is like the best microbiology magazine on the planet, bar none. And I don't say that because I'm the president of the society, but it is kick-ass. It's my favorite place to go and look at microbiology. So go have a look. Thank you. Oh, sorry. This is my son's band. I accidentally put this at the end of the last talk I gave. Still Euphoria, if you look it up and you want to scan it. He makes me put that on everything. He's 17 years old and this is about in North County with a drum kit and a truck. So, but they're pretty fun. Thank you.
Any question?
Well, this is because last week the single came out, so I had that. Yes.
Thanks for the talk. Really cool. You've worked on a lot of cool projects and initiatives, but what's the coolest one?
I sequenced President Obama's microbiome once. That was fun.
Oh.
So I got to spend 2.5 years building something called the National Microbiome Initiative with OSTP and became a presidential scientific advisor. And I wasn't a citizen at the time, but I worked for the Department of Energy as well, so that I had security clearance. So I got to do a lot of cool discussions with him and building up and then sequencing presidential poop. That was pretty cool. And then he was most annoyed because you can tell if somebody smokes based upon the bacterial community. So that was the one thing he didn't want Michelle to know about. Well, the fact he was still smoking at the time, but he was supposed to not be smoking. I'm like, you're the most photographed human being in the world right now, dude. So like, don't smoke. But can't stop people being people. But that was fun. I must admit, he was super intelligent. We did some stuff on the International Space Station as well, but that's just more annoying.
Any other question?
Has there been any work with engineering those microbes to be able to use the nutrients a little bit more effectively?
Yeah. So both we have a super cool system. Adam Feist here. Does anyone know Adam Feist? I love that guy. So he's got more energy than me, but he has something called ALBot. So you set the system running, and it's like that movie. There's a cool book. Anyone know what I'm talking about? The spaceman and the bacteria in the sun killing everything. Yes. So he uses adaptive laboratory evolution to evolve the bugs from the atmosphere of the planet, right? To be able to grow in a more nitrogenous, maybe, or carbon or oxygen-rich environment. Can't remember. Is it?
I think so.
Yeah. But so he's got a robot that does it, which is super cool. So you can force nature to evolve, right? And so that's what he's doing. We're also using dropout mutations, so not exactly adding functions, but taking functions away, which helps some of our bugs to be better at fighting off different pathogens. In agricultural systems, we have a large proposal we just put into USDA to go for that with Pythia, which is this freaking annoying little proteacean pest, which goes around wiping out large crops around the US. If we can solve that problem with a microbial solution, would save the US agricultural community like $175 billion. So they're thinking about giving us about 1.2 million to solve that. And I'm like, that's an order of magnitude wrong. But yeah, sure, why not? Engineering is incredibly important. I diss GMOs in the talk very, very slightly, right? GMOs are incredibly important, but there's several companies which just said we can solve everything with GMOs, right? And so they created their own super strain, which had lots of additions and modifications. Turned out that adding one bug with a bunch of functions into an agricultural system fails after about 2 growing seasons. because it can't compete in the soil system because it's not designed to compete in the soil system. It's designed to have functions which human beings thought were important. Biology is a hell of a lot more inherently complex. It's like human society, right? You got to figure out how to work with the problem, not work against it.
Time for maybe.
Or not.
Would it be a cross contamination if you bring a species of bacteria from one continent to another, even though it could improve the function?
Yeah, so that's the reason Rwanda, for example, in that commercialization effort, requested that we identify the strains that were identified originally in Brazil, but we identify them in Rwanda. Kagame wants Rwandan strains of these species. Just so happened that's totally feasible. So we have a consortium of 32 different bacterial species. They're found pretty much everywhere around the world. Why? Because if I sterilize the soil, like with a fire, right, and the surface is completely sterile now, within an hour, you'll have over 500,000 cells of bacteria landing from the air or from the rain onto that square foot, square inch, whatever, right? That smaller area will be bombarded with microbial life because the bacteria are being lifted up and transported through the atmosphere. You're breathing in right now. Every single one of you is emitting around 30 million bacteria from your bodies every hour, mostly through respiratory exhaling and from skin release. So you're breathing in each other's bacterial communities. And we found that people who spend more time together, especially if they physically interact, start having more microbial similarity. They actually share more microbes together. The soils are the same. It's a constant conveyor belt. The world is connected, right? But when they land in an environment and they can survive and thrive, they acquire mutations which make them kind of uniquely adapted or uniquely specific, endemic to that particular location. So whether or not it's important that we take Rwandan bacteria and use them in Rwanda, right? Rwanda being a human-made construct, right? Whether it's important that we do that or not, it's an interesting scientific problem, and also it's an interesting social problem. Because, you know, when I work with Sequan or La Jolla or any of the tribal communities out in East County, they don't want, you know, I once gave a talk to tribal council, and one guy just stood up and goes, "There's only three things in the world that I don't trust. That's white flour, white sugar, and white people. You're one of those, so no, I'm out." And I was like, "Cool, man. That's totally, I get it." But if your community is growing your own bacteria from your own soil, that's part of your heritage, right? So why not? And he's like, "Yeah, that I can live with," right? So that, to me, is the key piece. It's a social science question, first and foremost, and then a scientific endemic issue later on. Because I don't know what half of those acquired mutations do for one species of Syndovidium in that particular location. And I don't think I ever will, because I've got 400,000 species to figure out that problem for and not enough human beings to do it. Sorry, that was a bigger answer than you wanted, but you get where I'm coming from? Yeah. We can do it and we are doing it. Even the British want their own bacteria, you know. God save the king and put some microbes from British soil if there's any of it left.
I think we could all listen to him for another 6 or 7 or 8 days. Can't thank you enough, Dr. Gilbert. Wish we could go on, but I'm sure some people do need to get moving. Thank you so much. If you have not, are you sure?